bacteria that you do not care to eat. And so what I want to say, first of all, though, is that this is actually a relatively new field of research for my lab. I've been generally doing a lot of work on nervous system development. And so one of the things that I found as I started reading this is there are a couple of people's work that I really want to just focus on. Some of the foundational research that was important for guiding this study. It's certainly not a comprehensive list. But we were able to do some of this work largely because Jim Thomas did a screen for defecation mutants in the late 80s and published that in the early 90s. And Keith Nurke's lab has done a lot of work on intestinal physiology and trying to understand how the intestinal ion balance is maintained. Work from the Ausable, Garson, and Trauma labs were all instrumental in some of the techniques and things that they have developed for how worms are responding to pathogens, both genetically and physically. And then Eric Jorgensen's lab, who several years ago demonstrated that protons are actually released from the intestine onto the body wall muscles during defecation as part of understanding why some of these mutants are defecation defective. But really, this started a couple of years ago when I was working with a medicinal chemist at KU named Blake Peterson. And we had noticed, for reasons that I won't really go into at this point, that fluorescent molecules inside the intestine of C. elegans could exhibit a dynamic behavior. And we read that Keith Nurke's lab and others had used things like Oregon Green, which is weakly acid sensitive, to monitor the pH inside the C. elegans intestine. Unfortunately, those molecules are inactivated in the presence of acid. And so when you put them in acidic environments, they become non-fluorescent, which makes it more difficult to see some of these changes. So Blake's chemists started working on developing a series of analogs of rhodamine that could become activated in the presence of acid. So they developed this scaffold, which goes from a cyclic to a non-cyclic form with some regions on this ring structure that they could modify. And that by modifying these, they could change the pKa at which these rhodamine sensors would go from fluorescent to non-fluorescent and back and forth. And the one I'm going to talk about most today is this KR35, which has a pKa of about 3.5. So these were all published several years ago, and we were able to use them to actually monitor the fluorescence, or use this to monitor the pH inside the intestine of animals. I should point out that these are actually cell permeable. So if anyone is interested, you can get in touch with me about that. And some of these further neutral or more alkaline shifted pKa probes will show up inside, say, endosomes in other compartments within the cell that are slightly acidic. However, if we put this KR35 probe in the worm's just media, put it on the plate, we just sort of top dress it and let them crawl around on it, they will eat it. And then if we watch what happens, we can see that using a lookup table in which most fluorescent is red and less fluorescent is green, there is a dynamic wave in which the fluorescence moves throughout the intestine. And this happens approximately every 50 seconds and happens in animals that are well-fed and freely moving on the plate. When you anesthetize them or immobilize them, this behavior kind of goes away. But when we watched this many, many times, what we noticed was that during this sort of resting phase, when there was no, there were no dynamics, that there's a gradient from the anterior to the posterior, where we often see a more intense fluorescence in the posterior, and that this represents likely a more acidic pH than we find in the anterior part of the animal. Every time this cycles, we see a decrease in the fluorescence in the posterior with an increase in the anterior fluorescence, which we have now termed this maximum anterior transition. This then resolves where the entire intestine becomes neutral for a few seconds, and then it returns back to this resting state. Approximately the same time that we published this KR35 series of dyes, another group working with some ratiometric nanosensors were able to show that the pH is approximately 5.5 in the anterior and 3.5 in the posterior, but their approach did not demonstrate the same kind of dynamics that we saw. At least they didn't publish that. So we really got interested in trying to understand why the animals are doing this. So this twofold gradient of pH is representing a pretty significant energy investment of moving protons in and out of the intestinal lumen. So this cycle corresponded to the Defecation Motor Program that had been described a long time ago. So for those of you who haven't had the joy of sitting and watching worms poop, I recommend it as a very sort of Zen practice. But in this intercycle, worms have what we would observe as a pH with the lower pH in the posterior and a more neutral or slightly acidic pH in the anterior. At this time, when this posterior body contraction occurs, this is when we see the transition of our probe into the anterior. There is a relaxation period where we start to still see some anterior fluorescence. There is an anterior body contraction and then expulsion. So this was all described by Jim Thomas, and he was able to isolate mutants. So he isolated approximately 6 mutants that failed to undergo the PBOC and 6 mutants that had an absence of expulsion. So they didn't do this last step. Because of the correlation we found between those two, the DMP and this acidic wave, we decided to concentrate on some of these mutants and see what happened when we probed our mutants with those. Let me just tell you how this works, sorry. In the intestinal lumen, which is storing these protons, this seems to be generated by a series of either vacuolar ATPases or other proteins whose job it is to deposit protons into the lumen. In the, during the DMP, there is a calcium wave that travels from the anterior of the animal to the posterior of the animal that is dependent on the IP3 receptor and gap junction proteins like INX16 that allow the passage of calcium in this anterior to posterior wave. In the posterior of the animal, this appears to have a proton wave that goes from the lumen across the cytoplasm, where they are released on the pseudocoelomic face of the intestine and trigger body wall contraction through a ligand-gated ion channel called PBO5, for which protons are actually the ligand. Okay, so this was all work that was, this part of this work was done by Eric Jorgensen's lab. And so if protons are normally being evacuated from the lumen and being transited across, this doesn't really tell us why we have this posterior to anterior movement of the protons. So we examined some of these mutants, and 2 of them that were really relevant to us turned out to be from Jim's collection, PBO4 and PBO1. So both of these are mutants that lack the posterior body contraction, but are competent to do expulsion. So what we noticed was that in resting animals, so if we take a picture about 30 seconds before this maximum anterior transition, wild type animals have this gradient, and PBO4 animals are relatively normal. However, when the wild type animals are doing this maximum anterior transition, PBO4 mutants are unable to acidify this anterior part of their intestine. In contrast, PBO1 mutants exhibit the opposite phenotype. The interior is acidified. As far as we can tell by the fluorescence of our probe, it is more acidic than wild type animals, and this persists. So these animals we've never observed a dynamic wave in these animals. So what are these? So PBO4 is a sodium proton antiporter. So this does electroneutral exchange of hydrogen ions for sodium ions on the plasma membrane. And PBO1 is a EF hand protein that binds to calcium. It also, in a calcium-dependent manner, can regulate these NHX-like proteins or NHE proteins via a sequence on the cytoplasmic tail. So there are about 9 of these in C. elegans, and there's some evidence to believe that PBO1 and maybe other things can regulate the activity and localization of these channels. But again, because we had an opposite phenotype within them, we started to ask, well, could we use this to determine what is the consequence of the acidic wave? Why are the animals both generating this proton distribution, and why are they flipping that every 50 seconds or so? And we had sort of 2 main hypotheses. One was that it has something to do with digestion. So in our own bodies, when we're fasting, the pH in our stomach becomes acidic, and that acidic pH is important for the beginning of food digestion. So pepsin and other kinds of proteases that are acid activated are going to be more active in an acidic condition. when that food passes into your intestine, it is often buffered by things like bile acids and bicarbonate. The other, and sort of more perhaps colloquial discussion, is that we have acid in our stomach to kill pathogens that we might ingest accidentally while we're eating. So we didn't really have a favorite in this, but we figured that the pathogenic question was easier to address. We have seen a number of labs describe pathogens that C. elegans are, when exposed to, are detrimental to their health. So we hypothesized that because these animals had a more acidic lumen, that they would be resistant to pathogens, and these PBO4 mutants that cannot acidify the interior would be more susceptible. So we first tested this using Ephecalus. So Ephecalus is a pathogen for worms. It's not a very strong pathogen. So when you feed animals Ephecalus, they will die about twice as fast as they will when you feed them a laboratory strain like E. coli OP50. But we found our hypothesis was 100% wrong. So it turns out if you are a PBO1 mutant, you die about twice as fast on Ephecalus than you do if you are wild type or PBO4 mutant. Now, PBO1 mutants are relatively sick, but as far as we can measure, their lifespan doesn't seem to be particularly affected by that. So on this normal diet, they seem to have a relatively robust and normal lifespan. So one question is, well, maybe this is just specific to how the animal's responding to Ephecalus. So we tested a couple of other pathogens. We tested Staph aureus and Pseudomonas aeruginosa. Both of these are actually stronger pathogens. And in all cases, being deficient in PBO1 meant that you were more susceptible to these pathogens. We created a survival index where we basically took the ratio to set the mortality for each of these animals to, and 2 animals to each pathogen. And what we found is that for all of these pathogens, being deficient in PBO1 made you approximately twice as likely to die, or you died twice as fast in response to those pathogens. And that was not true on a non-pathogenic bacteria. So this didn't really tell us whether or not this was due to this pH. It turns out PBO2, which was also identified in Jim's screen, has been renamed AGL8. And we know that AGL8 is part of the innate immune system under other contexts. So to tell that, to differentiate that, we actually went ahead and added sodium bicarbonate to the worm's buffer, or we buffered the plates using sodium bicarbonate. Again, this is a physiological relevant buffer for our bodies. When we did this, if you are now PBO1 mutant, that completely reverted the susceptibility. So our relative mortality went back to being just like wild type. I should note that we found the best concentration to be about 25 millimolar. Once you got a whole lot higher than that in this particular assay, you started to get some diminishing returns. We went ahead and used our pH probe to document that actually the PBO1 intestine was becoming more neutral, and in fact that is the case. So the fluorescence intensity of our Kr35 dye is actually reduced in animals, PBO1 animals that are fed bicarbonate. So that led us to a question. Is the pH in the intestine modified by exposure to pathogens? So we basically, should mention first that we also did another experiment where we made double mutants of PBO1 and PBO4. So if we knock out PBO4 and PBO1 mutants, we basically revert the acidic phenotype, and that this phenotype now becomes more like the PBO4 single mutants, as does the pathogen susceptibility. So double mutants of the PBO1 and PBO4, in our hands at least, appear to be more like PBO4 than the PBO1 single mutants. And so this suggested that in both cases, whether you use a chemical approach or a genetic approach to reduce the pH inside the intestine, you're basically making the animals more resistant to the pathogen. So then we went in and tested whether or not exposure to pathogen changed the pH. So we grew animals on E. coli, fed them our pH probe, and then transitioned them onto plates that contained pathogens, either Ephecalus or Pseudomonas aeruginosa. And we found that as far as we could measure, that we saw a decrease in the intestinal fluorescence on these pathogens. We also saw a delay or a decrease, complete absence of the DMP on these pathogens, consistent with a failure in transporting protons across the cytoplasmic lumen, from the lumen to the pseudoslomic phase. This appeared to be less affected in these PBL1 mutants. So the ability to titrate the pH in these animals seemed to depend, at least in part, on the function of PBL1. So if we think about this, what is the purpose of having, or what are the mechanisms by which organisms are fighting pathogens in the innate immune system? There is a detection step. There is a transcriptional and or translational step where the animals are producing the orchestrated response. Some of those effectors turn out to be reactive oxygen species that Nick talked about, or antimicrobial peptides. These effectors are either, sort of bactericidal, so they kill them, or they're bacteriostatic, so they sort of manage the activity of these bacteria, and then the animal can eliminate them. So we actually went ahead and looked at some of these steps. So we took PBL1 mutants and used transgenes that have been developed in different labs, including Emily Trommel's, to look at the production of genes like IRG1 or the translational de-repression of beta-ZIP2. As far as we can tell, in those mutants using those transgenes, PBL1 mutants are able to do detection just fine. So we then went to the next step of this reactive oxygen species step. And we measured this using the Amplex Red assay, and Danielle Garson's lab was really helpful in getting us, helping us get this assay started. So if you take animals and grow them on E. coli and use this Amplex Red fluorescence indicator for detection of hydrogen peroxide, one of the key reactive oxygen species that are produced, you see a baseline amount. If you then challenge those animals with E. faecalis, you'll notice that wild-type animals and PBO4 animals are able to upregulate the amount of hydrogen peroxide they are producing, but PBO1 animals are not. If we went back in and then just acutely buffered that system with sodium bicarbonate, we were able to see a rescue of the production of hydrogen peroxide in the PBO1 mutants. I should also add that we tested mutants in E. faecalis that cannot produce hydrogen peroxide, and we demonstrated using those that the hydrogen peroxide was coming from the worms. One of the key ways that worms make hydrogen peroxide are by enzymes like BLI3. So we used RNAi to knock down BLI3, and we found that, again, we see a reduction in the production of hydrogen peroxide when challenged with E. faecalis, and that when we do this in PBO1 mutant animals, we did not see a further reduction, although honestly, we're probably working at a floor of hydrogen peroxide detection at this point. So one of the things, though, we did check was whether or not this caused an increase in the load of pathogens, either at 24 or 72 hours. And as far as we can tell using a very sort of like coarse assay at this point, is that we do not see a general increase in the amount of pathogens in PBO1 mutants. So it does not look like the infection is becoming systemic, nor does it look like it's having a change in the ability of the pathogens to grow inside the animal. So one of the things that we did notice, so we made a CRISPR-Cas9 knock-in of PBO1. So PBO1 appears to be almost entirely localized to the apical membrane of the intestine, although not really easy to see here in this single confocal slice. It appears to be more localized to the basolateral face in these posterior intestinal cells. So we've developed this model. So we believe that during this sort of resting phase, when the cycle is intermediate, there are resting calcium levels, and that is enabling PBO1 to be probably in an off position, and that PBO1 is on, especially in the posterior of the animal. And that's allowing protons to be deposited into the lumen. Now, we don't think that this is the only mechanism by which protons are introduced into the lumen, but we at least think this is one of the ways that that is occurring. We also know from other work that PBO4 is on this basolateral membrane, and it is likely off in this condition. During the DMP, PBO1 is probably responding to the increased cytosolic calcium levels. We believe that this is a negative regulator of PBO4, and I'll explain that in a second, but that is now basically allowing the flow of hydrogen ions into the pseudocoelomic, or into the cytoplasm, and release by the basolateral PBO4 onto the body wall muscles. Again, we think that PBO1 is a negative regulator of PBO4 because of the genetics. So when you knock out PBO1, you see a PBO4-dependent increase in intestinal pH. In the consequence of infection, though, we believe that you need to turn off PBO4 by increasing calcium levels. This has been demonstrated in a number of different epidermal wounding and pathogen-mediated events that calcium levels increase cytosolically, and that pH, or that allows a neutralization of the pH in the intestine, and that there's a deposition, and probably in an aquaporin-1 dependent manner, of hydrogen peroxide. However, in PBO1 mutants, where you cannot neutralize the pH, hydrogen peroxide is either destabilized or production is lost. And that this neutralization, or that this increased acidity, is detrimental to the normal response. Okay. I should just point out that our pH results are consistent with a recently published series of experiments from pigs that is from a cystic fibrosis model, where the experimenters were able to demonstrate that the air surface liquid of pigs with a CFTR mutant mutation is acidic, and that acidic air surface liquid has a reduced capability of killing Pseudomonas aeruginosa pathogens. They were able to rescue that entirely by buffering that air surface liquid with bicarbonate. And then they were able to show that there are specific antimicrobial peptides that have a lower efficacy under lower pH conditions. So a couple of our questions are, for the future directions, what is the pH-dependent mechanism in the intestine that is mediating the susceptibility to the pathogens? Is it directly via BLY3? Are there other hydrogen peroxide producing converting enzymes that are pH dependent? Is it really down to the level of antimicrobial peptides? Does neutralizing the lumen of the intestine acidify the cytoplasm? So there's some work from yeast to suggest that acidifying the cytoplasm can invoke specific kinds of transcriptional responses. Do other pH generating proteins in the development of this pH gradient depend on PBO1? And then, as Nick just talked about, there are a bunch of different pathogens that C. elegans can encounter. And we know from transcriptional studies that there are different transcriptional programs that can be activated by this. So the question is, are there other infections that are pH independent? We don't know that yet for certain, but we have definitely started to look at animals. As Nick had mentioned, we do know from different species of C. elegans that show different kinds of susceptibility to pathogens. So we started to ask whether or not they're pH dependent as well. So I'm just going to stop there and thank a lot of the people who did the work. In my lab, a lot of this work was done by Patrick Lansden, who's a postdoc in the lab. This was a collaboration with Josie Chandler at KU and her former postdoc, Saida Benamart, did a lot of the work as well. It was started as a collaboration with Blake Peterson, where Aaron Bender and Zach Wojcik helped us develop the probe and do some of the early tests in the PBO-dependent mutants. And I'd like to thank a lot of people for funding. And with that, I will stop and take questions.
All right. Thank you for a very good talk. We already have one question from Javier. And if anyone wants to virtually raise their hand, click on participants and raise your hand. So the question from Javier is like, could the bacterial pathogen be more pathogenic at low pH because they use the proton to uptake nutrients from the lumen?
Yeah, so that's a really great question. We've tried to think about ways in which we could would acidically primed pathogens be more detrimental to worms that are not PBO1 mutants. We haven't found a good way to test that, but certainly that's possible. I just read a paper, I think this came out in PLOS Genetics just recently, that there is a pH-dependent effect on E. coli in terms of cell division. And I'm not a bacterial physiologist, but I could certainly imagine that there are other pH-dependent events on bacteria that would cause them to be more pathogenic. All we can say for now is that a broad class of bacterial pathogens appear to have the same consequence in worms, right? So that approximately doubles the time in which PBO1 mutants will die in a pH-dependent manner. So if there is an effect on bacteria, it's pretty global.
All right.
Great question.
I have a question. Maybe you said it and I missed it. But how do you follow a single worm for like the fluorescence of like a full cycle? are they stuck in a microfluidic chamber or?
No, we're just doing this on plates. As I mentioned, if we put them in a microfluidic chamber, so when you stress them, they tend to stop the behavior. You'll see they stop defecating too. So this is just following them around on plates.
All right.
Yep.
Okay, so we have a question from Manish Shamoli.
Mm-hmm.
Great talk. Have you used this probe to check leaky gut phenotypes? Can leaky gut modulate intestinal pH?
That's a great question. And in fact, when we tried to publish this, there was a lot of questions about whether or not the pathogens were damaging the gut epithelial barrier. And as far as we can tell, in this case, this is sort of the opposite of what you're asking, but we don't see an increase in epithelial barrier, or we don't see a decrease in epithelial barrier function in the PBO1 mutants. So being hyperacidic, if I can use that term, doesn't seem to cause damage that we can tell. Whether or not the animals with a leaky gut phenotype would change intestinal pH remains to be seen. Since we've published this, I've actually gotten a number of people interested in looking at their mutants with our pH probe. And if anyone has a really good set of mutants to suggest, I would love to test that out. Yeah.
All right. Is there any more questions? Otherwise, you can still ask question on our Slack channel. I think I've seen you there. So
yeah, please, if you have any questions or feedback, as I mentioned, this is really kind of a new field for my lab, and so we are open to ideas.
All right. So thank you very much to you and Nick for like 2 great talks this morning.
Yeah, thank you. Thank you for organizing.
For depending where you are. And we'll have another session next week. I'm going to send a new email soon for update everyone about that. All right. Thank you very much.